So we've covered the details of the lymphatic system. Now let's look into how we defend ourselves against the pathogens that may actually be found in the lymph. The pathogens are environmental agents that are capable of producing disease. These include things like infectious organisms, toxic chemicals, radiation, so snake bites, bacterial infections, or cancers. There are 3 lines of defenses against these pathogens. The first line is our external barriers, the skin and the mucous membranes. They're impenetrable to most pathogens, thus their integrity is really important to defend us against them. Our second line of defense is the non-specific defense mechanisms. There are several different ones, and those are the content of this chapter. We'll look at how leukocytes, macrophages, antimicrobial proteins, immune surveillance, and inflammation and fever work. These are effective against a broad range of pathogens, and they're present from birth, and they work even against pathogens that the body has never experienced before. In the third line of defense, we involve the immune system. And the immune system not only defeats the pathogen, but leaves the body with a memory of that pathogen. So in the future, it can defeat it much faster. In nonspecific resistance, we see the systems are guarding equally against a broad range of pathogens. The effectiveness does not depend on any prior exposure. We'll see the skin and the mucous membranes. We'll explore how leukocytes and macrophages work, and some antimicrobial proteins. We'll also examine immune surveillance, inflammation, and fever. Immunity is specific defense because it results from prior exposure to the pathogen, and it usually provides future protection only against that particular pathogen. That'll be the content of our next section. So let's begin by looking at the external barriers. The skin makes it mechanically difficult for microorganisms to enter the body. There's that tough layer of keratin in the dead cells in the top of the integumentary layer, and it's too dry and nutrient poor to support any kind of microbial growth. We also secrete peptides that kill these microbes by creating holes in their membranes. We'll examine those later on in this section, as well as an acid mantle. As we sweat, we produce lactic acid, and that itself inhibits bacterial growth. Our mucous membranes that line the digestive, respiratory, urinary, and reproductive tracts are open to the exterior, and the mucus in the mucous membranes physically entraps microbes. Mucus, tears, and saliva also contain lysozyme. This is an enzyme that destroys bacteria by dissolving their cell walls. So we're fairly well protected from the external environment. We'll also see that beneath the epithelia is areolar tissue. And the areolar tissue has a ground substance in it. And this ground substance contains a really large glycosaminoglycan that's called hyaluronic acid. Hyaluronic acid gives the areolar tissue a very viscous consistency, and that makes it difficult for any pathogens to pass through. However, some pathogens actually make an enzyme called hyaluronidase. This enzyme makes hyaluronic acid much less viscous. In the venom of snake bites, there's often hyaluronidase, and that's how their toxins make it through this subepithelial areolar tissue barrier. Now, if the microbes get past this physical barrier of the skin and the mucous membranes, then they'll be attacked by phagocytic cells. The phagocytic cells have a voracious appetite for foreign matter. These phagocytes are leukocytes and macrophages. As we learned in chapter 18, there are 5 different types of leukocytes: the neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Let's begin by looking at the neutrophils. Neutrophils wander around in connective tissue killing bacteria. They phagocytose them and then digest them. They also produce a cloud of bacterial chemicals. This cloud of bacterial chemicals creates a killing zone. When the neutrophil detects bacteria, its lysosomes migrate to the cell surface and degranulate. That is, they discharge their enzymes into the tissue fluid. This causes a respiratory burst, in which the neutrophil rapidly absorbs oxygen and converts it into a series of toxic chemicals. This cloud of chemicals kills way more bacteria than the phagocytosis. Unfortunately, though, it also kills the neutrophil. So in this case, he sort of acts like a suicide bomber. The Eosinophils are found especially in the mucous membranes. They stand guard against parasites and allergens and other pathogens. So we'll find them especially concentrated in areas of allergy and parasitic infections. They kill tapeworms and roundworms by producing superoxide, hydrogen peroxide, and other toxic proteins. They also promote the action of basophils and mast cells, as well as phagocytosing antigen and antibody complexes that are floating around. They also limit the action of histamine and other inflammatory chemicals that, if they were left unchecked, could cause a lot of tissue damage. So basophils actually call other leukocytes to the site of pathogen infection. They do this in a few different ways. First of all, they secrete leukotrienes that activate and attract the neutrophils and eosinophils to the site. Second of all, they secrete histamine. Histamine is a vasodilator, which increases blood flow, and thus it speeds the delivery of leukocytes to the area because there's an increase in the blood flow. They also secrete heparin, and heparin inhibits the formation of blood clots. If blood clots were to form in that area too soon, it would impede the mobility of the leukocytes and thus impede their activity in cleaning up the site. Mast cells also secrete these substances. They're a type of connective tissue cell that are very similar to basophils. Now we have leukocytes. There are three basic categories of leukocytes that we've already mentioned. There are T cells, B cells, and natural killer cells. About 80% of lymphocytes are T cells, 15% are B cells, and 5% are the natural killer cells. The roles of these lymphocytes are too diverse to address here, but we'll definitely describe them later in our section on immune surveillance. So monocytes are white blood cells that emigrate from the blood into the connective tissue, and then there they transform into macrophages. And as we know, macrophages gobble things up. There's a whole system of macrophages. It's all of the body's avidly phagocytic cells, except for the leukocytes. Some of those macrophages are wandering macrophages, where they actively wander around and seek out pathogens. They're widely distributed in the loose connective tissues of our bodies. Other macrophages are fixed. They phagocytose only the pathogens that come to them. These are cells like the reticular cells. There are also some specialized ones in specific regions of the body. For example, microglia we see in the central nervous system, gobbling up all the pathogens around the neurons. And alveolar macrophages can be found in the lungs, and hepatic macrophages can be seen in the liver. So in addition to all of these specialized leukocytes and macrophages, there are antimicrobial proteins that will help us protect against pathogens. There are multiple types of proteins that inhibit microbial reproduction and provide short-term nonspecific resistance to pathogenic bacteria and viruses. We'll describe two families of antimicrobial proteins here, the interferons and the complement system. Interferons are secreted by cells that have been infected by viruses. They're basically the dying words. They don't benefit the cell that's infected themselves, but it puts the word out there that they have been infected. This alerts the neighboring cells to protect them from becoming infected. These dying words or secretions, proteins, bind to the surface receptors of the neighboring cells and activate second messenger systems within them. It alerts the cells to synthesize various proteins to defend it against the infection. It breaks down viral genes and thus prevents the viruses from replicating. The signal also activates natural killer cells and macrophages. Once these are activated, they come to the site and they destroy the infected cell before it can liberate a whole swarm of newly replicated viruses. We'll see that the interferons confer resistance not only to virus-infected cells, but to cancer. The activated natural killer cells can also destroy malignant cancer cells. The second mechanism we'll talk about is the complement system. The complement system is a group of 30 or more globular proteins. They make powerful contributions to both nonspecific resistance and specific immunity. Their pathways of action are fairly complex, so we'll just sort of scrape the surface here. They're synthesized mainly by the liver, and they circulate in the blood in an inactive form. They're activated by the presence of a pathogen. This activates 4 methods of pathogen destruction. First of all, there's inflammation, immune clearance, phagocytosis, and then cytolysis. We'll look at each of these in detail. This occurs through 3 routes of activation. There's a classical pathway, an alternative pathway, and a lectin pathway. You can see each of these different pathways here, and your text goes into more detail. However, I won't hold you responsible for all the details of these activations. The thing to note is the C3 protein through either pathway will be dissociated into C3a or C3b. And either C3a will mediate some of the responses, like the inflammatory response, or C3b will mediate the immune clearance, phagocytosis, and cytolysis response. I only bring these names up because you're going to see them come about as we discuss inflammation, immune clearance, phagocytosis, and cytolysis. So in the mechanism of inflammation, we'll see that C3a stimulates all the mast cells and the basophils. They secrete histamine and other inflammatory chemicals, which activates and attracts the neutrophils and the macrophages, and it speeds the pathogen destruction in the inflammation. You can see this pathway right here. In immune clearance, we'll see C3b having an action. It binds with antigen-antibody complexes to red blood cells. These red blood cells circulate through the liver and spleen, and the macrophages in those organs strip off and destroy these antigen-antibody complexes and leave the red blood cells unharmed. This is the principal means that we use to clear foreign antigens from the bloodstream. Immune clearance. Binding with these antigen-antibody complexes that are stuck to red blood cells. And in the liver and the spleen, we strip them off. It's simple as that. Again, you can see these pathways here in immune clearance. The diagram, I think, is kind of a nice way to summarize all of the actions. Again, don't get too much into the details of what C3a and C3b is, just that it can be activated in 3 different ways in order to have these antimicrobial proteins have their effect. So then we need to gobble up invading pathogens. That's in the phagocytosis portion. Neutrophils and macrophages cannot phagocytose naked bacteria, viruses, and other pathogens because they don't recognize them as foreign. So C3b will assist them through the process of opsonization. That is, they coat the microbial cells, and that serves as binding sites for the phagocyte attachment. Basically, it's like sprinkling on a little salt. It makes the foreign cell more appetizing to the neutrophils and the macrophages. Their role in cytolysis is breaking apart the cell, is that C3b splits other complement proteins. So it binds to the enemy cell and attracts more complement proteins. So here we can see a whole number of different complement proteins. Again, don't get into the details of it, but basically C3b will attract all these other complement proteins to the enemy, and it'll form a hole in the cell. And literally, the fluids and electrolytes leak out of the cell and the cell ruptures. So again, we were just looking at the complement system. And in the complement system, we're looking at a load of different proteins that can help protect us against invading pathogens. The way they can do this is by activating 4 different means of pathogen destruction. They're involved in activating inflammation, immune clearance, phagocytosis, and cytolysis. 4 different ways to destroy pathogens. We saw that one of these complement proteins, we see that each of these 4 means of destruction can be activated by several different pathways. Don't worry about these pathways. Just recognize that they activate these different means of destruction. We've got inflammation, immune clearance, removing stuff from the blood, phagocytosis, gobbling up the pathogens, cytolysis, destroying the pathogens by blowing holes in their sides. Okay, so that's a lot of material. This would be a great time to pause and do a little drawing. First of all, identify the 3 different lines of defense. Next, outline all the different leukocytes and what their functions are and how they accomplish those functions. And third, think about how the antimicrobial proteins work. There were 2 classes, the interferons and the complement system. How do interferons work? And then outline the complement system. Again, not too much detail. The complement system proteins are a bunch of proteins. That's all you need to know. And they have effects on moderating these 4 different responses that can get rid of pathogens: inflammation, immune clearance, phagocytosis, and cytolysis. How do they do that? Then we'll move on in our next section to look at immune surveillance, fever, and inflammation.